Hello everyone, we're Group 33 and we're presenting the computational analysis of prostate adenocarcinoma. Our research question is that by performing differential expression analysis on gene expression data as well as mutation data, can we deduce a relationship between the pathways obtained and the genes mutated? What are the trends and factors that affect protein expression in the patient data? Our project goal is to identify subtypes and frequently mutated genes of prostate adenocarcinoma using differential gene expression, survival analysis, hierarchical clustering, and unsupervised machine learning models to deduce a relationship and correlation between tumor progression and patient survival rate. We obtained our raw data sets from the TCGA database, including the RNA-seq data, mutation data, and patient clinical data. Through our analysis, we aim to reveal novel molecular features and mutated genes to identify potential biomarkers for prostate cancer. First, we analyzed our clinical patient data, and then we performed a gene expression analysis of our RNA-seq data, and then we conducted a survival analysis of the following features. We then analyzed the mutation data through clustering to find relevant signaling pathways. We also plotted a survival analysis plot based on the patient survival status, which showed that 5 patients did not survive.
We start our analysis on the RNA-seq data by applying the Ward clustering method. After, we conduct a differential expression analysis using DE-seq, where we obtain the following 80 upregulated genes and 5 downregulated genes. After conducting the DE analysis on our upregulated genes, we found the top 5 pathways that were identified as highly enriched by the upregulated genes. These were the cardiac muscle contraction, olfactory transduction, calcium signaling pathway, oxidative phosphorylation, and gastric acid secretion. For our downregulated genes, after our DE analysis, we found that there are 5 pathways that are associated with the genes. These are protein processing in endoplasmic reticulum, lysosome, RNA transport, ubiquitin, mediated proteolysis, and N-glycan biosynthesis. Let's analyze the pathway analysis results of our data. The downregulated genes enrich the pathways that are mainly responsible for protein processing, transport, and proteolysis. For each downregulated gene pathway, the proteins that were most significantly expressed in red were identified. We found that the overexpression of N-glycan in PCA, or prostate cancer, is a possible biomarker for diagnosis. Also, MGA-T4C, the differentially expressed protein of N-glycan biosynthesis pathway, poses a deletion risk and is significantly overexpressed in metastatic and localized prostate cancer. The expression of HYAL1 from the lysosome pathway can impact the behavior of stromal cells, which suggests that cell-cell communication via exosomes due to elevated HYAL expression promotes prostate cancer progression. Similar analysis was performed on upregulated gene pathways. It was found that IP3R, a protein common to the gastric and calcium pathway, regulates the calcium flux into the cell and thus controls cell apoptosis, indicating that it might be a biomarker for PCA. TRPML1 in calcium pathway is found to play a role in mediating cellular bioenergetics and is found to be overexpressed in PCA, and its downregulation results in autophagy, leading to cell damage and DNA arrest. M3 protein in gastric pathway plays a role in MAPK signaling and regulation of actin cytoskeleton. These two factors are thought to play an important role in signal transduction by M3 in prostate cancer. In the oxidative phosphorylation pathway, protein NDU-FV2 participates in androgen regulation and are thought to thus play a role as a biomarker for PCA.
The survival analysis was also performed on the mutation data by appending all clinical patient data to mutations using the patient ID. By then doing that, we were able to conduct a survival analysis for variant type, where we found a deletion variant type to have a significant p-value and to be statistically more deadly than other variant types. We clustered then all of the clinical patient data according to which of the top 20 mutations were shown for that patient. With those clusters, we then performed another survival analysis on that clinical patient data using the clusters obtained from the mutation data. There was a high p-value indicating no statistical significance unless the high p-value is attributed to the small sample size. Then it can be concluded that patients dying from prostate cancer quickly had all similar mutations in the top 20 genes. As seen in the cluster dendrogram, patients were clustered according to their similarity in the top 20 genes that were mutated. Then, using those clusters, a DE-seq analysis was conducted, and from that differential expression analysis, KEGG pathways could be obtained. The top 5 upregulated pathways from that analysis were vascular smooth muscle contraction, vagus regulation of the actin cytoskeleton, focal adhesion, and the MAPK signal pathway. On this slide, we see the upregulation of vascular smooth muscle contraction. The important takeaway from this slide is MHC right there on the side being induced in upregulation. On this slide, we look at vagus. Again, the only important takeaway from this slide really is to look at that MHC1 expression there being induced. On this slide, we see our results from downregulation. Although no p-values were significant, we did see it suppress collagen in order to suppress immune signal production.
Based on our pathway analysis of mutation data, the statistically significant pathway was the vascular smooth muscle contraction pathway, which regulated MHC production, and also MHC1 production was regulated in the phagosome pathway. Looking at other pathways, we see more regulation of other proteins that help reduce cellular apoptosis and create an accumulation of misfolded proteins.
Continuing this discussion, in tight junctions as well as in focal adhesions as well as cell ECM comes into play. From these, we see that the cell is having increased expression and interaction with its environment. We also see the ERK5 pathway regulated by a transcription factor. However, that's likely the result of clinical applications to stop cancer inflammation. Additionally, there's an upregulated protein in the endoplasmic reticulum, which is HSP72, which is responsible for negatively regulating apoptosis. Having a defect on this gene means less cell death will occur.
We found a few limitations to our analysis. Only 5 people died from prostate cancer. The small data set made it hard to get significant p-values, and the raw data set was highly unstructured, which required a lot of pre-processing. Also, the large amount of NA values in the clinical patient data made survival analysis very difficult. We found that no major trends were noticed between the patient survival, tumor stage, and tumor subtypes in our survival analysis. For our gene expression data, we found that downregulated and upregulated gene pathways revealed the various proteins that could act as potential biomarkers for prostate cancer diagnosis. Also, for our mutation data, we found that the analysis of our regulated gene pathways revealed only one significant pathway, which was the vascular smooth muscle contraction, which inhibits immune response through MHC1 production. Protein processing in the endoplasmic reticulum was the common downregulated pathway in both gene expression and mutation data analysis. The MAPK signaling pathway was obtained as a result of upregulated genes in both mutation and gene expression data and is known to play a significant role in signal transduction in prostate cancer. Thus, our results can relate back to our research question of finding potential biomarkers and signaling pathways for analysis of prostate cancer, which can also be validated by literature findings. Thus, this concludes our presentation. Thank you for listening.